And first of all, first things first, nganong kailangan man nato sa diyan ng carbohydrates? Well, typically, carbohydrates kay isa siya sa mga primary metabolite na importante kaayo sa atong life. Okay? First of all, ang carbohydrates, siya ang responsible nga anong naay unique identity ang atong dugo. We have four types of blood. Okay? Naay blood type A, naay blood type B, AB, and type O. And ang unique feature aning each of these blood types is actually the sugar component. Like for example, si blood type A, blood type B, and blood type O, same sila sugar units kani, same sila ani, but nag-differ lang sila sa isa ka sugar unit. Like for example, kani si blood type A, ang iyang sugar unit, ang third sugar unit niya sa iyang structure sa antigen is N-acetyl glucosamine. Whereas kaning si blood type B, ang iyang third sugar unit is galactose. Kanin siya. Si blood type O, wala siya third nga sugar unit. So mo ni ang differences nila, and it's all because of the carbohydrate component. Next up is ang carbohydrates kay ginagamit siya sa cell para mag-recognize og outer pathogens and some toxins. Okay? So, like for example, toxins, viruses, bacteria, before sila maka-invade sa sulod sa cell and makabuhat sila sa ilang purpose, kailangan nila muagi sa cellular membrane. Ang kanang cellular membrane, it's composed of a variety of carbohydrates. Okay? Daghan kanang carbohydrates sa surface sa cell. And ang analogy is, for example, kung ang kaning toxin, viruses, bacteria is like estudyante nga high school, before siya makasulod sa eskwelahan, kailangan siya moagi sa gate and kailangan siya i-check sa guard. And ang guard mo ng mga carbohydrates, kintahay. So, importante kayo ang carbohydrates for recognition sa mga pathogens. Kaya kung wala sila, basta-basta na lang ni makasulod ning mga viruses, toxins, and bacteria. And here is an evidence nga ang cell surface is daghan siya carbohydrates. And this one is called a glycocalyx. Ang kaning glycocalyx, it's a very complex kanang murag gelatinous type of layering outside sa cell wherein ka-compose siya of various carbohydrates, glycolipids, glycoproteins. It's a complex aggregation of those carbohydrates, okay? And even the derivatives. So importante kaayo nga kanang sadihan ginato ang carbohydrates. And of course, we all agree nga all cells are sugar coated, either animal cell or plant cell. So, unsa pa na siya nga cell, all of it have carbohydrates. And also, carbohydrates make up biopolymers. Biopolymers such as proteins and nucleic acids. Kani siya nga mga biomolecules kay part sa iyang structure is a sugar unit. So, importante po ning carbohydrates para ma-achieve ang 100% functionality aning mga nucleic acids and proteins like glycoproteins and stuff like that. So part po ni siya sa central dogma kaning nucleic acids and proteins, and without the carbohydrates it cannot make up its structure. And of course ang cell dili mag-function. Ang kaibahan lang gid sa proteins and carbohydrates is that ang proteins ginakuan siya kanang sa tawag ani gina-produce siya sa nucleus, sa genes itself. Okay? Whereas ang carbohydrates dili siya gikan sa kuan, dili siya gikan sa cell mismo, sa nucleus sa cell. Ang carbohydrates nga naa sa cell surface gikan na siya sa mga sugars nga gikaon nato sa atong pagkaon, na metabolize siya, gina-absorb siya sa cell and gina-metabolize siya sulod sa cell, and ang end product is gina-release siya sa cell para himuon siyang layering sa iyang surface, and it becomes another sugar. So, it becomes a sialic acid, to be particular. So, a glucose, gikan sa atong pagkaon, sulod siya sa cell, and pag human og metabolize dito sulod sa cell, i-release siya sa cell para himuon siyang covering or skin sa cell in a form of sialic acid. So that's how the chemistry works in the cellular level. This is a table that describes the general role and function of carbohydrates. Number one, carbohydrates is used for energy storage. Like for example, ang mga tao kung dili siya mokaon og anything for about let's say 2 weeks, mabuhi gyapon siya, nagyapon siya kusog. That's because ang atong cells gina-use ang energy from the carbohydrate storage para mabuhi gyapon ta, para makalihok pa ta and napatay energy. And those carbohydrates nga na ay energy sources are glucose, starch, and glycogen. Glycogen particularly makita ni siya sa atong muscles. Mao na nga kung gamiton na siya sa atong cells or sa atong lawas ang kaning energy aning glycogens or i-metabolize siya muna magniwang ta kay gakakuan atong muscle mass. And of course, structural integrity, as what I've said, carbohydrates act as barrier. It acts as a membrane. It's like a skin for the cells. And for the plants, of course, we have cellulose na makuha nato in a form of a very complex like polymer of sugars. And chitin, of course, chitin is an exoskeleton nga present sa mga crustaceans like shrimp, crabs, and the rest, and some sort of like the same similar kinds of animals like lobsters, and also chondroitin. Okay, makita siya sa cartilage. So of course, ang kaning carbohydrates, it is part of genetic information. So nucleic acids kay naa siya phosphate group, naa siya sugar, naa siya ATP, and naa siya sugar unit. Of course, like RNA and DNA, it has a sugar unit in its structure. And of course, for cellular recognition and identity, just like the blood type groups A, B, AB, and O. So these are the commodity chemicals nga pwede nato makuha gikan sa carbohydrate sources, like for cellulose makagama tag paper, cotton, linen. And from cellulose acetate, this is a derivatized form of cellulose wherein ang OH groups sa cellulose kay gihimong acetate groups. Pwede ta maka-produce og yarns, disposable filters, and upholstery. Of course, the most common sugars like fruit sugar, fructose, and sucrose from sugarcane, pwede ta maka-generate of food products such as cane and beet sugar, and also corn syrup nga very important sa food industry. So this is the chemistry now of carbohydrates. So what is carbohydrate? Ang carbohydrate, it is simply a polyhydroxyl aldehyde or ketone. Like in basic organic chemistry, this is the functional group aldehyde, right? RCHO. O pwede po nato siya nga sulaton nga RC double bond OH. This is the aldehyde. And ang ketone naman is this one. Mo na siya yung functional group. Polyhydroxyl aldehydes because daghan kay kag hydroxyl groups. Poly, from the word poly. So, like for example, this one. 1, 2, 3, 4, 5. 5 ka hydroxyl groups. That's why it's polyhydroxyl aldehyde because ang functional group present is aldehyde. Okay. And of course, napod siya yung mga derivatives such as DNA, deoxyribose. So, it's pag may kag deoxy, ang isa ka hydroxyl unit is replaced with H. Like for example, this one, mahimo siyang H. You know, it becomes now a deoxy sugar. So ang carbohydrates, gi-categorize siya into two: monosaccharides and complex saccharides. Unahan nato si monosaccharide. Si monosaccharide is dili siya ma-split into simpler forms. Okay? It's now the monomer of all carbohydrates. So it cannot be split into simple molecules. How will you know that a monosaccharide is a monosaccharide kung na kay unknown nga substance? Do hydrolysis. Kung dili gali siya ma-hydrolyze, then it's a monosaccharide. Okay? So monosaccharides, for example, if this is a sugar unit, a monosaccharide unit, no? Dili ni siya ma-hydrolyze. No hydrolysis happens. It cannot be um, turned into a simpler unit. And also, we have complex saccharides. Complex saccharides, there are three types: disaccharide, oligosaccharide, and polysaccharide. Okay? Ang disaccharide na siya'y duha ka monosaccharide. Ang oligosaccharide, ang pinaka-simple nga oligosaccharide is what we call a trisaccharide. This is the simplest oligosaccharide. And ang oligosaccharide, it is comprised of 3 to 25 sugar units. Ang kaning oligosaccharide. And ang pinaka-complex sa ilahatanan is the polysaccharide, such as cellulose, glycogen, na chitin, mga polysaccharide na. So, ang polysaccharides mostly is composed of 26 to 1 million sugar units. Okay. And disaccharide, of course, from the di word or prefix, duha lang ka monomers. Okay. This is sugar 1 and sugar 2. Notice na there is in these complex saccharides na ay linkage. Okay? Na ay bond. Mo na siya ang ga-connect sa mga monomers. And ang tawag ana saya is called the glycosidic linkage. Or na other terms ane. It can be also called as glycosidic bond or acetal linkage. Mo na siya. So, let's go back to history. Sa unang panahon, ang tawag gid sa mga monosaccharides is hydrate of carbon because naa siya nakita sa mga chemists nga naa siya formula nga consistent ani na general formula CnH2n H2ON. Okay? That's why it came from French word hydrate de carbone. Ambot saan siya pag-pronounce basta mo ni siya French word. So it's hydrate of carbon. And then in the early 19th century, ang mga sugars na common gakakita sa nature is gipangalan siya based kung asa siya gikan or asa ang iyang source. Like for example, glucose, it's named after the grapes nga source niya. And of course, fructose nga gikan sa fruits, any kinds of fruits. And sucrose, it's named after sugarcane. Very common nga source sa sucrose. And in 1866, which is part of the 19th century, si August Kekule, nagpropose siya names for glucose and fructose. And ang iyang proposal is glucose is named as dextrose, whereas fructose is named as levulose. Okay? Dextrose because kung ang glucose daw itunawon siya sa tubig or it becomes an aqueous solution, it will turn the plane of polarized light to the right in the polarimeter. Okay? Kung familiar, kung gitudluan mo ani sa organic chemistry ninyo, so you will know what I'm talking about. So, kung yung turn to the right nga pag-turn sa plane of polarized light, it's clockwise, okay, inside the polarimeter. And si fructose naman, levulose, kay kung imuan siya aqueous solution, ang plane of polarized light is anti-clockwise ang turning inside the polarimeter. Okay? And from 1880, which is still a part of the 19th century, naitulo ka individuals nga nagpropose og systematic naming for carbohydrates. Sila si Fisher, Van Hoff, and si Lebel. So sila ang nag-adapt sa current nga ginagamit sa IUPAC rules in naming carbohydrates karon. And even gi-consider nila ang stereochemistry sa mga chiral centers in a given sugar molecule. Okay. So, giunsa man pag-form ang carbohydrates. Actually, carbohydrate is a primary metabolite. Ginasynthesize siya naturally by nature, even sa plants, bacteria, and all living things. Okay? Except the humans. Okay? Di ta makaproduce of our own carbohydrates. Okay? So, kailangan na siya i-take. And ang formation sa carbohydrates mechanistically is parehos siya sa mechanism sa ginatawag natong na acylloin condensation. It's an organic reaction. Okay, nga mahimo lang siya sa lab. Sa acetylene condensation, naa kay homocoupling. Homocoupling of two ester molecules. Mag-coupling silang duha to form an acetylene compound. An acetylene is a hydroxy ketone. Why hydroxy ketone? Because hydroxy because it has a hydroxyl group, and a ketone because it has a ketone functionality. And it needs a sodium metal and xylene as a solvent, and it also requires heat. So before ma-produce ang hydroxyketone, mudaan mo na siya through two intermediates, a diketone or an indiol in a form of sodium salt. I will not discuss the mechanism because radical type ang mechanism ani. And the important thing or key takeaway is that same ang mechanism sa formation of carbohydrates with the acetylene condensation. So in nature, ang pag-form sa sugar is nagsugod gyud na siya sa carbon and water. Si carbon and water undergoes via enzymatic processes and mahimo siyang formaldehyde-like unit. Okay? CH2O. It becomes N, meaning kung 1 siya, isa lang siya ka-formaldehyde unit. Pwede po siya 2, 3, and so on. So, kaning formaldehyde units, kung nakay duha ka buok, they will condense together. Okay? To form this hydroxy aldehyde here. So, ang mechanism sa pag-condense nilang duha is somewhat similar sa acetylene condensation. Okay? Ang mechanism. So, it becomes an acetylene type compound. It's just that aldehyde lang ang isa ka functionality. Kung naa kay formaldehyde unit, remember nga gikan siya sa carbon and water, no? Kung naa kay formaldehyde unit na mo-condense with the aldehyde functionality, it becomes a ketose or a ketose sugar. Kung naa kay formaldehyde nga mo-condense with an alcohol unit sa acetylene type molecule or structure here, it becomes an aldose or an aldehydo sugar. So inana ang yang mechanism of formation. So acyclic derivatives of carbohydrates kay naay duha ka types, aldoses and ketoses. So from the word itself, makaingon ta nga aldoses contains an aldehyde functionality and ang ketoses contains a carbonyl or a ketone functionality. And ang suffix sa aldose is -ose, whereas sa ketose, -ulose. Mo na systematic way of naming aldoses and ketoses. Like for example, kani 1, 2, 3. Tulo siya ka buok carbons, so this becomes a tetrose. By the way, same rin siya nga molecule. So kani siya 1, 2, 3, 4, upat ka buok carbon atoms, this becomes tetrose. And this one 1, 2, 3, 4, 5, 6, this is hexose. So ang ketose naman, diba, remember, mapansin ninyo, aldose na siya aldehyde functionalities. Ang ketose, on the other hand, naa siya ketone functionality. So, kani siya, mga ketoses ni siya. And ang pag-name, ang prefix sa name is describes the number of carbon atoms, whereas the suffix is the -ulose. Okay? So, this becomes a sorry, this is not tetrose but a triose. Sorry about this. So this becomes a tetrulose. Okay, tetrulose because it has 1, 2, 3, 4 carbon atoms. This one is 1, 2, 3, 4, 5. This is pentulose. This one is 1, 2, 3, 4, 5, 6 hexulose. So inana siya pag-identify kung naay -ulose na suffix, that's a ketose. But kung -ose lang, that's an aldose. Sorry sa akong cat. So, ang aldose, pwede siya ma-derivatize or refunctionalize. For example, you have a given aldose molecule here, aldose structure. Remember sa organic chemistry, Natay different kinds of oxidation levels from alcohols to aldehydes, ketones, aldehydes and ketones, and carboxylic acids. So suppose this is an alcohol, right? This is an aldehyde. This is a carboxylic acid. Alcohol, aldehyde, carboxylic acid. In going to the right, here, this is oxidation. And going to the left, this is reduction. And ang symbol for reduction in chemistry is H, tapos for oxidation will be O. Okay? Mao ni akong gamiton nga symbol is symbol. So, ang aldose pwede siya mahimong alditol. Alditol. Okay? Here, alditol. Kung imong i-reduce ang kaning aldehyde functionality, this is an aldehyde into an alcohol functionality, diba? From aldehyde to alcohol, it's reduction. You create an alditol. Alditols could also be converted into aldonic acids. So how? By oxidation. If you oxidize the alcohol functionality, any alcohol functionality, as long as it's in the terminal position, terminal meaning sa pinaka-last or pinaka-kilid, it could be this or it could be this. Okay? But isa lang ka-alcohol functionality. Kung imo siyang i-oxidize, mahimo siyang aldonic acid, and ang iyang functional group now becomes a carboxylic acid, and this is oxidation. And aldonic acid, it can be converted into uronic acid. And particularly, you need to oxidize and reduce two functionalities. One is carboxylic acid. Kung imong i-reduce, you have an aldehyde here. And ang kanin namang terminal nga hydroxyl group, if you oxidize it into carboxylic acid, this is now a uronic acid. This one. Uronic acid can be converted into aldaric acid. And for aldaric acid is ang terminal ends niya are both carboxylic acids. Here, carboxylic acids na siya. 1 and 2. And isa lang ka functionality from the uronic acid ang imuhang i-oxidize. From aldehyde to carboxylic acid, this is an oxidation. So this is the interconversion between aldoses, okay, via redox processes. And here is the family tree for aldoses. Diba? Aldoses, naa siya aldehyde functionality. And all of this stuff right here, naa siya aldehyde functionality as the core functional group. Okay? Here. So this family tree describes the common aldoses. Okay? So this one, since 3 carbon atoms siya, 1, 2, 3, this is a triose group. Triose, isa lang siya, glyceraldehyde. This one, threose and erythrose, is tetrose group. This one is the pentose group. And this one is the hexose. And sa nature, mostly ang gina-produced good is the hexose sugars because of stability issues. And later on, I will discuss how important stability is sugars. So in here, naay various names. These are trivial names. Wala gitay choice but to familiarize ourselves with the trivial names and also the structure sa positioning sa mga hydroxyl groups. And for example, threose, sa threose ang iyahang hydroxyl groups naka-trans, whereas sa erythrose is naka-cis ang iyahang relationship. And naapoy other convention to name this aldoses, and that is by indicating the configuration, which is D here, Of course, in nature, naturally, sugars are in D configuration. And the other one is by abbreviating the name. And sa pag-abbreviate sa name, kailangan lang og first three letters sa full name. Just like lexose, LYX, xylose, XYL, arabinose, ARA, RIB for ribose, talose for TAL for talose, gal for galactose, and so on and so forth. But there is an exception to this convention. It's glucose. This is an exception. Di ba, ingon ko ganina nga ang pangalan, kung i-abbreviate siya, is first three letters. But why is it that glucose is GLC? Kung sundon tong convention, dapat GLU. But ang ginagamit sa carbohydrate chemist is GLC. Why? What's the reason? Ang rason is, nakauna naman gud ang mga peptide chemist og actually set up og systematized naming of peptides. And if you're familiar with an amino acid called glutamic acid is symbolized as GLU. So, to avoid confusion sa mga peptide and carbohydrate chemist, nag-giveaway na lang ang mga carbohydrate chemist, and mas nauna man si glutamic acid good sa convention of naming. So, that's why for glucose, para ma-avoid ang confusion, gihimo na lang siyang GLC. And also, napod tayo mga ketoses. And ang common sa ilang tanan is the ketone functionality. Here. This is a tetrose. This is a, sorry, not tetrose, but tetrulose. 1, 2, 3, 4. This one is pentulose. And this group is hexulose. Okay. So, kani pong mga names nga indicated in green, kailangan po ni siya i-familiarize and memorize on. D-fructose, psychose. But there is another convention. You indicate the number of carbon atoms. Okay? So, in here, it's a pentulose unit because, for example, ang cellulose, gitawag siya as D-threopentulose. It's because ang kaning kani siya, it's somewhat like similar to the threose. Diba? Ang iyahang na siya CH2OH, ang OH na yan nasa right, ang OH nasa left, tapos na siya carbonyl functionality. It's just that ketone lang dire. So that's why it's called D-threopentulose. Okay? The same thing with this. It's called D-erythropentulose because it kind of like similar in terms of structure sa D-erythrose. Okay. D-tagatose. Kung imo lang tan-aw ni nga functionality, it's somewhat like similar to the structure of the lactose here. So that's why it's called D-lactopentulose. And of course, for sorbose, like arabino, D-fructose, for example, it can be named as D-arabinopentulose because this particular moiety in the structure is somewhat like the structure of arabinose. So that's another convention for naming ketosis. So far, naka-discuss na ako ang mga structure sa aldoses and ketoses. So this brings us to a summary about the requirements for carbohydrates. Like, unsaon man nimo pagkabalo nga ang kani siya nga molecule can be considered as a carbohydrate. So how do you do it? How do you categorize them as a carbohydrate? Number one requirement for a molecule to be a carbohydrate is that naa siya dapat naa siya secondary alcohol. Okay? Of course, like this. Like this, naa siya, diba? This is a secondary alcohol, for example, because ang kani nga carbon is naa siya duha ka R groups, diba? So it's a secondary alcohol. And another thing is na dapat siya primary alcohol. It has a primary alcohol because this is a primary alcohol. Okay? Kaya ang carbon na connected sa OH, nalang siya isa ka R group. That's why it's primary. And also, dapat na siya carbonyl functionality. Aldehyde or ketone. That is why kuan siya kanang aldose or ketose, so because of the carbonyl functionality. So the kaupat is dapat na siya at least 3 carbons. So ang pinaka simple nga sugar is the glyceraldehyde. This one kaya na siya at least 3 carbon atoms. So these are the 4 requirements for carbohydrates nga matawag nimo siya nga carbohydrate ni nga molecule. Basta naa siya ani ma-meet niya nga mga requirements. Okay. So in carbohydrate chemistry naatay 5 different representations for hexoses. Sorry. One is the Mills projection, Haworth, chair, Fisher, and symbol. Ang kaning symbol, ginagamit din siya sa mga biologists because biologists and other related disciplines did not care about the structure. But chemists in particular care about the structure because important sa ato ang stability sa molecule. Okay? And naputay Mills projection and Haworth projection. Mo ni siyang sorry kay akong earring kay saba kayo. Yuki. Ang Mills projection and Haworth projection, mao ni siyang kasagara gina show sa mga biochemistry textbooks, but it does not really tell you the stability and energy of the molecule. So it's just showing you the orientation of the hydroxyl groups like this. Hydroxyl groups. Haworth and Mills. Mills projection is like naa ka sa top niya, nagtan-aw ka sa ubos, nakita nimo ning kuan. Nakita nimo ni siya kaning nga molecule. Kung mayroon kag hardly shaded bond, it's leaning towards you, and kung hashed, it is away from you. So palayo siya sa imo or nasa sa likod. And let's just say na this is a plane of the paper. Okay? This is the plane of the paper. Ang kaning entire structure ani, kaning ring, nani siya within the plane of the paper. Na. And kaning mga bonds is ga-protrude siya out basta heavily shaded. Kapag gawa siya sa plane of paper, meaning gaatubang siya sa imo. Okay? Pero kung kaning hashed, kaning broken lines, na siya sa likod actually. Inana ang yang 3D representation sa Mills. So, ang kaning wiggle nga bond, it only tells you na ang stereochemistry aning OH group is not emphasized, no? Dili siya gina-describe kung R ba siya or S. It could be either R, it could be either S. Mo na gina-indicate sa wiggle nga bond. So, in other words, ang OH group, it could be protruding towards you, or it could be oriented away from you. So, dili na to ma-identify kung asa ang iyang orientation. So, that's the wiggle bond. And sa Haworth naman, kung diri ka magtanaw sa kilid, diri, sample, diri imong mata, kung naka-flat bitaw ang kaning papel, niya na imong mata diri, mo nang Haworth projection. Ang gipahigda ra nimo ang Mills projection, and gitawag gitanaw nimo siya nga pahigda sa munang Haworth. And ang kaning sa Haworth, ang kaning bonds nga heavily shaded, mo ni siya ang sa top, whereas kaning broken line sa Mills projection sa Haworth, it's at the bottom. Okay. And ang Fisher projection, it's an acyclic structure. Okay? But pwede po siya macyclic, actually. but it does not tell you the relative energy and stability of the sugar molecule. Okay? And ang Fisher projection, ang importante ra sa iyaha is that makita nimo ang stereochemistry. Makita nimo ang stereochemistry sa individual carbon atoms. Okay? Mao nang importante sa Fisher projection. But naturally, ang sugars exist as a cyclic structure with a chair conformation. So mo gini ang pinaka-exact nga structure sa sugars because it will tell you the energy and stability of the molecule. Okay? Like this, glucose. Glucose is the most stable, pinaka-stable siya nga sugar molecule. Why? Because it does not have any stable siya in the sense na wala siya, there is no, nakuha na akong pencil. Wala siya 1,3 diaxial interaction. Ang diaxial interaction is kung kani ang sugar molecule is carbon 1, 2, 3, 4, 5, H. Kung na kay OH groups dire and napog kay OH nga axial, this will be steric interaction. This is called a 1,3 diaxial interaction. Okay. So, usbon ako, ang chair conformation ang pinaka-perfect nga ga-demonstrate sa structure sa carbohydrates. And not the Mills, not the Hayward, not the Fisher, and even not the symbol. So, Fisher projection. Ang Fisher projection, sayon ra siya tan-awon actually kay acyclic manggod ang iyang portray, and this one is called a perspective projection. Means a perspective projection is ang kaning heavily shaded bonds right here kay gaatubang siya sa imuha, mura bitaw siya ginani mura siya gaatubang sa imo, di lang ako siya ma-drawing. Basta gaatubang siya sa imo kani. Tapos kaning duha ka bonds nga broken kay gaatubang siya palayo sa imo. Nana siya pag kanang identify or paglantaw. And of course, sa Fisher projection, pag number sa carbon atoms is number 1 gyud dapat ang most oxidized carbon. And sa structure sa aldoses kay number 1 ang kaning CHO. Okay? Kay siya man ang pinaka-oxidized. Aldehyde oxidation level man siya. And sa unang panahon, nagtuwa ang mga chemist nga kung nagi kay D sugar, before na ko na siya i-discuss, no. By the way, ang mga Fisher projections, it also shows you ang isa pod ka importance niya kay it shows you the mirror image, no. Madali ra nimo identify kung mirror image ba ang sugar relative to another sugar. Just like for example, this one. This is a mirror image of this one. Kung nakay mirror in between, mo na yung reflection. Okay? So, ang sugars is naa siya duha ka configuration. The D configuration, D for dextrorotatory. L for levorotatory. Okay. O saan man pag-identify kung dextro siya or levorotatory based on the Fisher projection? Simple lang, pangitaon lang nimo ang penultimate carbon. O saan man ang penultimate carbon? Sinsya, saba kaya akong iring. Ang penultimate carbon, siya ang carbon nga naay nga pinakalayo, siya ang chiral nga carbon nga furthest away from the most oxidized carbon. So in this structure, ang pinaka-oxidized nga carbon is this one kay aldehyde man siya, no. Aldehyde. Ang pinakalayo nga chiral carbon, these are chiral carbons kani. Chiral ni siya. This one is not chiral kani. Because naa siya duha ka H, similar groups. So it's an achiral carbon. Ang pinakalayo nga chiral carbon is kani siya. So, dire mag-determine ang D and L configuration. Okay? Kung ang OH group is naa siya sa right, it's a dextrorotatory. Kung ang OH group sa penultimate carbon is naa sa left, it's levorotatory. Okay. And sa unang panahon, ang mga chemists nagtuo sila nga pag D sugar kay positive ang optical activity. This is optical activity. Optical activity is mo na siya ang pag-rotate bitaw sa kanang kung i-subject nimo siya kung ang sugar imo nimo siya aqueous solution tapos i-subject nimo siya sa polarimeter. So kung mag-turn siya, kung i-turn niya ang plane polarized light to the clockwise direction, positive. Pag negative is anti-clockwise direction. So nagtuo sila sa una nga pag D sugar is clockwise direction inside the polarimeter, mo nang plus ang iyahang equivalent and ang L is negative, but actually no, dili siya independent ni silang duha. It doesn't necessarily mean nga pag D sugar is positive ang sign. No, napoy mga D sugars nga negative ang sign. The same thing for L. L is not always negative sign. It could also be positive sign. So independent ang plus minus nga symbolism and the DL configuration. Okay. And ang problem manggod pod sa Fischer projection is that you can always rotate the molecule. Okay? And rotation happens when you have saturated bonds, like sp3 bonds ba? There is free rotation in sp3 bonds. So magdepende po just sa groups nga present, no? But in organic chemistry, fundamental OChem, sp3 bonds can rotate. Na siya free rotation. And kung here, kung lakay di configuration, for example, kung ibo siya i-rotate clockwise ang molecule, like the CHO is transferred to here, OH is here, CH2OH is here, and H is here. Shh! Sorry, ako i-rig, saba kayo. So, i-rotate mo siya 90 degrees, na 90 degrees ang rotation. May mo siyang L configuration. And kung i-rotate pagid siya nimo further 90 degrees, mahimo siyang D configuration again, this one. So retention is equal to double inversion. Kailangan nimo siya makuan. Kailangan nimo siya i-doubly invert aron makabalik ka sa original nga configuration sa imong sugar, no? So here, proteins versus carbohydrates. Ang proteins is composed of monomers called amino acids, ba? Ang amino acid na siya inaning general formula, amino acid. And also carbohydrates, ang iyang monomer is the monosaccharide. Ang amino acid naa siya negatively charged N here and naa siya positively charged N. This one. By the way, this is carbon atom, okay? Sa amino acid, naa siya electrophile. Napod siya nucleophile. Here. So, in a given amino acid, like this, naa kay electrophilic end, napod kay nucleophilic end. Okay. And kung mag-combine kag duha ka amino acid, kintahay, na. So, kani, combine mo siya, mo siyang na, diba? So this bond is what we call as a peptide bond or amide bond. Ang carbohydrates inanipod na yung same gyapon siya sa amino acid. But it's just that sa carbohydrates or monosaccharides, daghan kay siyang nucleophilic sites. Like say for example, this one, if this is a sugar molecule, let's just say when gi-represent niya ning structure, no? So this is a nucleophilic site. The negative charge here represents the OH kay nucleophiles man ang OH. Okay? and ang positive site, which is the carbonyl. In a monosaccharide structure, mo ni siyang electrophile, ang aldehyde or ang ketone in terms kung nakay ketose. Ang nucleophilic end is actually this one. This is the nucleophilic end. Pwede po ni siya kani, pwede po kani ang nucleophilic end, pwede po kani, pwede po kani. Sorry, not this one. Ani, mga nucleophilic sites na sila, OH. So, kung na kay other sugar molecule, kintahay, na negative, 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 positive. Okay? So, combine mo siya, same nagyapon siya sa ano, sa carbohydrate, sa amino acid nga na then negative, 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 negative. Same nagyapon siya. Pwede siya mo react dire, pwede po siya mo react dire. So, lahi-lahi nga orientation. Ang akong gi-indicate ani is when the last OH or ang pinaka furthest OH is mag-react sa electrophilic inani nga reaction ba. So, in nature, 99% of carbohydrate exists as a cyclic structure, and a cyclic carbohydrates mo cyclize gina siya. Okay? Cyclize gina as always because of stability issues. Kay kani magong chain or a cyclic structure is since all of the bonds here are sp3, naa siya free rotation, so maka-adapt siya, maka-twist siya, maka-rotate, so ma-adapt siya conformation, and kung maabot niya or ma-achieve niya itong conformation nga mura siya cyclic type gani na ayha siya mag-cyclize. So how do carbohydrates cyclize? Ang pinaka-fundamental reason is reaction of carbonyl compounds. So, very important kaayo nga makabalo ta sa chemistry of carbonyl compounds. And in particular, it's the nucleophilic substitution at carbonyl with the loss of carbonyl oxygen. Okay. So to demonstrate this, suppose you have a carbonyl. Let's just say an aldehyde and a nucleophile. Okay. Of course, the nucleophile will react here. Well, let's just say it's acid catalyzed. Okay. First, the aldehyde gets activated. Right? Becomes activated and then i-attack siya sa nucleophile. And gani siyang reaction. Ang sp2, this is an sp2 carbonyl, it becomes an sp3 bond sa intermediate pa lang. So, it's nucleophile and then H. Now it becomes sp3. Right? Ang problema magodalig, this is called a tetrahedral intermediate. Okay. Ang problema magodaling tetrahedral intermediate is it's unstable. Unstable siya. So, suppose that the OH gets acidified. Okay? Napakay remaining acid catalyst. So, ang tendency is sorry, not this one. Suppose that you have H leaving group. Okay. This is an activated form na sa carbonyl compound. Suppose you have a leaving group here. It's now a carbonyl derivative na living group. Okay? And when the nucleophile attacks, Lg is leaving group. It's nucleophile and then OH. Okay? Especially kung nakakay ka ng sorry, this should not be acid condition. Sorry. Na kung nakay leaving group, eject out ang leaving group. Diba? And now it becomes a ketone. Okay? Substitution reaction. Mo ni naka disadvantage sa tetrahedral intermediate kay it is too unstable. Okay? So pwede siya nga mag-kick out og leaving group if ever your substrate or starting material has a very good leaving group such as hydronium ion, oxonium ion, tapos chloride or chlorine atom, for example, or iodine. Na. So, mo ang basic sa nucleophilic substitution at carbonyl atom. So, ang formation sa cyclization, by the way, ang cyclization sa carbohydrates is, ang iyahang fundamental mechanism is hemiacetal formation. Okay? Sa maning hemiacetal. Ang hemiacetal, suppose that this is R1, R2. This is a carbon atom at the center. Ang hemiacetal is naa siya alkoxy and hydroxy. Okay? Hydroxy. Hemiacetal formation, it could be catalyzed under acid condition. It could push through under acid condition. So, acid catalyzed. So, kung sa acid catalyzed conditions, kung na kay, say, aldehyde, right? And you protonate it to make it activated. By the way, kung activated ang himong aldehyde, nagpasabot na nga it becomes more electrophilic. More electrophilic. Susceptible na siya for attack sa nucleophile. So now you have R. Oh, let's just say the nucleophile is an alcohol because ang acetal kay reaction man siya. Ang hemiacetal is a na form siya when an aldehyde or a carbonyl compound reacts with an alcohol. Okay, so this is now an OR. Okay, so H. So this is a hemiacetal. Kung base catalyzed, ang una mahitabo is that the alcohol, gets deprotonated first in the presence of a base, strong base. This is okay, so this becomes now a very kung sa acid catalyzed, ang carbonyl or ang aldehyde, it becomes more electrophilic. Sa base catalyzed, lahi. Ang alcohol becomes more nucleophilic. And bisag walay activation sa carbonyl, maka-react yapon siya because it's more nucleophilic now. And kaning oxygen is it will protonate itself kay naa kay base di ba dire, and the base will abstract a proton, so it becomes now a conjugate acid. So ang conjugate acid, it will be deprotonated. So you now have a hemiacetal. These two groups are hemiacetals. Okay? So, mo na ang basis sa formation sa cyclization. Mo na ang basis sa cyclization sa sugars. Okay. Mo na siya nga mechanism ang inasunod nila. And also, napoy acetal formation. Ang acetal formation naman, it only uh, undergoes via an acid catalysis. Only acid catalyzed. Kaya kailangan magug ka sa acetal formation, kailangan na kay living group. Ang carbonyl oxygen, kailangan nimo siya himong effective nga living group. So kung nakay basic, it doesn't become a no kung nakay base sa reaction, it does not come off as a living group ang kanang carbonyl oxygen. Okay? So here, kung nakay course, nakay aldehyde, imo siya siyang himong more electrophilic by protonating the carbonyl oxygen so that it becomes more electrophilic here. So it will be attacked since acetal man siya, so the nucleophile will be an alcohol. Alcohol will attack here, and then OR. Okay, so kani siya napag second nga catalysis may tabo. Another, this oxygen here from the tetrahedral intermediate will be protonated further, and this becomes a good leaving group now. So that kung naay another alcohol here, second alcohol molecule will attack, and this come off as water. Minus H plus, then minus water. So this now becomes, this is an acetal. Ang acetal is naa siya duha ka alkoxy groups. So ang hemiacetal, OH, OR. Ang acetal is duha ka OR groups. Okay. Yan ni ang formation gagakahitabo sa disaccharide siya. Disaccharide formation. Okay? Acetal siya. Mura siya acetal type mechanism. Whereas si hemiacetal is cyclization sa monosaccharide. Okay. So, kung i-demonstrate nato ang cyclization sa carbohydrates, inaani siya. So, suppose you have here a glucose molecule, D-glucose, right? Kung ato siyang i-drawing in a, like, pahiga. OH, 6, 5, 4, 3, 2, 1. So this is the another structural representation of this structure, no? So this could also orient in a way na mura siya ganang cyclic type, pero acyclic siya. This is 1, 2, 3, 4, 5, 6. This is 1, 2, 3, 4, 5, 6. Visual projection, kaya pa na siya. And this molecule, this one, this could be, in nature, napod tayo mga acid catalyst actually sa nature. Napod tayo mga bases. Nature, in particular, ang mga bases is in the form of bicarbonate. Okay? And also some enzymes have this like ability to catalyze such reactions. Okay, it could either protonate this one, it could either make it activated, this carbonyl, diba? Same siya sa hemiacetal and acetal mechanism. Or pwede po nga ang base, it will deprotonate first the OH at carbon 5, and after such deprotonation, this becomes O minus and will attack this. So either way is possible. It could be base catalyzed or acid catalyzed. Okay. So here, ipakita lang ako ang neutral mechanism. Nga walay catalysis, but actually in nature, naagi na siya catalyst in the form of enzymes. So here, lone pairs will attack. cyclization happens. Minus H plus. Na. So, ina niyang cyclization mechanism sa carbohydrates from acyclic to cyclic form. And ginasunod niya ang acetal or hemiacetal mechanism, either acid or base catalyzed nga mechanism. So, introduction to monosaccharides. Ang monosaccharides, common nga monosaccharides as what I mentioned earlier is hexoses. So, naapod siya mga stereocenters, no? And a stereocenter is given by a formula 2n, wherein 10 is the number of chiral carbons. So, mo siya ang general formula to identify how many stereoisomers are present in one molecule. Okay? So, here are the common monosaccharides. Here, 6 of them with their structures in meals and chair conformation. Okay. So, in here, napod tayo stereochemical relationships for monosaccharides. Epimers. Epimers kay mo na sila mga isomers nga differ lang sa isa ka chiral carbon. Like for example, if this is our baseline or if this is our base sugar, which is the glucose here, galactose is a C4 epimer of glucose. Why? Because same sila tanan og stereochemistry and even functionality nga nasa aldehyde groups. But at carbon 4, ga-differ sila because sa glucose, it's on the right. The hydroxyl group is on the right, but sa galactose, na siya sa left. That's why it's called a C4 epimer of glucose. Ang mannose naman, it's a C2 epimer of glucose because they differ at the chirality is different at carbon number 2. Okay? Sa glucose, nasa right, whereas sa mannose, nasa left. And the rest are the same. Ang anomers kay ma-identify lang nimo siya kung ang sugar is in cyclic form. Easy to identify kung cyclic form, okay? So suppose you have here a hypothetical sugar ring, okay? Chair conformation. Kung ang OH, 'di ba sa chair conformation kay kung sa chair conformation, here, this is the banana maning orientation sa bonds. Na, this is equatorial, axial, equatorial, axial, equatorial, axial, equatorial, axial, axial, equatorial, equatorial, axial. So in here, the oxygen, the hydroxyl, kung ang hydroxyl group gali is naa sa axial position. Axial. Ang tawag na niya is the alpha anomer. Kung ang OH is na siya sa equatorial position, it's a beta anomer. By the way, it's a cyclic structure na ata ginatawag nga anomeric, ito ginatawag nga anomeric carbon. And ang anomeric carbon kay it's like an acetal. Okay? Remember the acetal or the hemiacetal. Mo na siya ang carbon. Like, di ba sa this is the acetal and this is the hemiacetal. Ang chiral carbon kani anomeric carbon rather. This is a anomeric carbon. So in this structure, ang anomeric carbon is kanang indicated sa green. And to identify the anomeric carbon, siya ang naay duha ka oxygen nga nakaban sa iya ha. Mao nang anomeric carbon. Siya lang ang nag-iisang carbon nga naay duha ka oxygen. And the anomeric carbon is the most electrophilic carbon. in the sugar molecule. Mo na nga ang naay gakahitabo nga disaccharide, trisaccharide because of the electrophilicity sa anomeric carbon. Diri gina mostly ang bond gakahitabo kung nakay disaccharide. Okay, diri na siya mostly sa anomeric carbon gakahitabo ang chemistry, the formation sa polysaccharides. Okay. And also like kung Hayward projection naman, this will be the anomeric carbon. Kung ang OH is nasa top, na this is beta. But kung ang OH is nasa bottom, this will be alpha anomer. And of course, napoy mutar-rotation kitabo. Ang mutar-rotation is phenomenon siya nga kung ang sugar gali, example naka-sugar, kani siya sugar ni siya, and you mix it with in a glass of water, for example, it now becomes an aqueous solution, diba? Kung solid siya ang sugar, it adopts a specific type of configuration, for example, alpha. But kung i-dissolve mo siya sa tubig or mo naman siyang solution, it could exist as a mixture of alpha and beta. Mo na mutar-rotation. So in here, as a mutar-rotation, ang cyclic move daan mo na siya sa acyclic and then mubalik siya pagka-cyclic. Okay. So in here, the alpha anomer, kung i-dissolve mo siya sa tubig, no? Ma-break ang kaning band nga dire, ma-break siya. It becomes now as acyclic form. Katong Fisher projection nga akong ipakita kaniha. Or open chain or acyclic. And at the same time, it will cyclize back again to form another anomer. At this time, it becomes a beta. Okay, and this happens in equilibrium. Magsigir ni siya padulong diri or padulong diri. Inanagay kitabo kung ang sugar is in aqueous solution. Okay. Like example, kung nakay pure anomer, ang imong gisubject sa polarimeter, ang iyang optical specific rotation kay 112.2 degrees. Kung imo siya i-dissolve sa tubig, it will be in open chain first and then cyclize again to generate the pure beta anomer. So from alpha, gidissolve nimo siya sa tubig, nahimo siyang open chain, and in such a specific kind of conformation, mo cyclize back siya again, generating a beta anomer. And ang disadvantage, o ang not a disadvantage, but ang bantayanan is that, for example, kung ang kaning alpha anomer, imo siya i-dissolve sa tubig and gibiyaan nimo siya for long periods of time, let's just say 48 hours. Lusagdan na rin nimo siya. Sorry, not 48 hours, but let's just say 1 hour. Gipasagdan mo siya for 1 hour. For 1 hour, nahimo siyang beta anomer. But ang iyang specific rotation, it now becomes 18.7 degrees. Okay? But, kung ang kaning alpha anomer, gidissolve mo siya sa tubig for, let's just say, 48 hours, kanang dugay jud kaayo, this 18.7 will eventually become 52.6. And this is the highest, mo na ni siyang pinaka-high nga optical rotation nga ma-achieve aning beta anomer. Meaning ani kung ang beta anomer nga imong ka-observe is naa siya 52.6 nga rotation, specific rotation is pasabot na 100% ani is beta anomer. Okay. So here, to simply describe the mutar-rotation phenomenon, suppose you have a straight chain sugar. Okay. So this will adapt a cyclic like structure, right? And this will cyclize to form the, by the way, this is in equilibrium. Okay, this is the anomeric carbon. And since nasa equatorial ang kaninga OH, this is a beta anomer. Okay? Suppose nga, since as what I've said, sp3 bonds can rotate, right? So suppose you are rotating this 180 degrees. Okay. Mahimo in such a way nga mahimo siyang nani. Kay gi-rotate man nato siya. Na instead of carbonyl oxygen pointing up at 180 degrees, pointing down ang carbonyl oxygen. Okay? So if this happens, still the same phenomenon, adapting the cyclic like open chair form. But this time, nasa down ang carbonyl oxygen and H ang up. So if this happens, if cyclization happens, okay, sorry. Now, this is an alpha anomer because this is the anomeric carbon, right? Carbon, and ang OH is eggshell. So this is an alpha anomer. So inani ang mutarotation. Kung nakay pure alpha anomer, it will open into an open chair form like this and go to this and cyclize and rotate. Gaka-rotate siya because it's in solution now. So mo rotate ang bond. So paagi ani mag-cyclize na pud again, and it now becomes a beta anomer. And likewise from beta anomer, it comes through this mechanism. There will be free rotation of the bonds when you have aqueous solution of the molecule of sugar. Okay. So inani ang mechanism that describes the mutarotation. And mag-different din ang ilang specific rotation. So, another disaccharide. Ang disaccharide, it's composed of the, like on my introductory slide, right? And the saccharide, siya duha ka monosaccharide, diba? Just like this. Sucrose is the most common disaccharide apart from lactose and maltose. So si sucrose naa siya alpha D-glucose and beta D-glucose. Alpha because with respect to the anomeric carbon, right? Ang O, which is OH before siya naghimong disaccharide, is eggshell. That's why it's alpha. Beta D-fructose because the OH, the anomeric OH is it's oh sorry, this is anomeric OH, and this is beta relative to the structure of fructose. Okay. So ang disaccharide na siya systematic naming pod. Ang glucose is gagahimo siyang alpha D glucopyranoside. This is the systematic naming, and fructose becomes beta D fructopyranoside. Sorry, not pyranoside. Furanoside. Since this can be collectively called as glycoside. Glyco from the word glyco meaning sugars, okay? So, why pyranoside? Why furanoside? Ang reason is gikan ni siya sa heterocyclic compounds nga pyran, and furan. Ang pyran is 6-membered ring. Ang furan is 5-membered ring. And sugars, such as glucose, are 6-membered ring, right? 1, 2, 3, 4, 5, 6. Fructose is 5-membered ring. 1, 2, 3, 4, 5. Okay. In naming, sa pag-name sa disaccharide, systematically, dapat una gid ang it should be non-reducing end and then reducing end. Kung saan man mo pagkabalo nga non-reducing? Not end, but sugar. Non-reducing sugar and reducing sugar. Okay. Kung saan man mo pagkabalo nga non-reducing, let's just say unit. Non-reducing sugar unit or followed by reducing sugar unit. Okay. Pagkabalo nimo, o sa mo pagkabalo is that ang non-reducing sugar unit is na siya acetal. Diba? Acetal has this formula. So, if this is the anomeric center or carbon, this will be the OR group and this is another OR group relative to this anomeric carbon. So, morning acetal. Kung ang reducing sugar unit is naa siya hemiacetal. Hemiacetal is OH, OR. Remember this structure or formula. So, relative to this anomeric carbon here, naa kay OH and a very big OR. Sorry. This is an OR. Doesn't matter what the R is, but it only tells you like the rest of the molecule. So, sa pag-name sa disaccharide, na po siya trivial name such as this one. This is maltose composed of 2 glucose units. Sa pag-name is, diba, this, kung wala pa siya collect, this is supposed to be alpha D glucopyridoside. But in naming, kung disaccharide na siya, this is still as a monosaccharide, no? But kung disaccharide, since this is the non-reducing part or end, this will become alpha D glucopyranosyl. Ang ide gilisan nimo YL. So that's the how to name it. That's how you name it. That's why a non-reducing end or non-reducing sugar unit, it's alpha D glucopyranosyl dash 1 arrow 4 D glucopyranose. Okay. So, why alpha? It's because relative to this anomeric carbon here, ang OH supposed to be HOH man na siya. Naa siya sa bottom, so it's an alpha anomer. Whereas ang D-glucopyranose, it should be well kay wiggling bond man siya, so meaning wala naka-indicate ang position sa OH good exactly kung top or bottom. That's why wala siya gi-describe diri as beta or alpha. Gidiretso siya as D-glucopyranose. But kung top galing OH, this should be beta D glucopyranose. Okay? Glucopyranose because it's glucopyranose or glucopyranoside, it's the same thing. Okay. So, kung inani nga system or convention, dapat na siya dash parenthesis 1 for 1 because related to the non-reducing sugar unit, it's at carbon 1. Nag-start ang glycosidic linkage. This is the glycosidic linkage and connected siya sa carbon 4 sa reducing sugar unit. Okay. This is a 1,4 glycosidic linkage or glycosidic bond. And napoy shortcut pag name. Gamiton nimo ang abbreviation like GLC for glucose, okay? But kung inani nga convention, wala na ning dash. That's why it's now alpha D glucose. Nga naka-connect siya from 1, carbon 1 up to 4. Carbon 4 of the reducing sugar unit, which is this one, and then D glucose. Or pwede po siya nga instead of arrow, comma. Okay. practice. Here, this is beta-lactose, this is maltose. So, kung ato siyang i-name as this, by the way, this is chain. Not chair. Sorry, chair. This is Haworth projection. By the way, kani siya is it's not a carbon in between rings. It's just showing you the orientation of the original OH groups kung katong wala pa sila nag-react. Okay. So, to name this one, it should be, since this is a galactose, okay? Galactose unit, and this is glucose unit. You see, the galactose is a non-reducing. Kay naa siya OR, and then this is another OR. It's a non-reducing sugar unit. Si glucose naman, it's a reducing sugar unit because relative to the carbon, anomeric carbon, na siya OR and then OH. It's a hemiacetal. Hemiacetal, and this is a full acetal. Okay? So to name this will be beta because it's equatorial here. Okay? And also here it's equatorial. So it's gonna be beta D configuration, galactopyranosyl dash, and this is carbon 1, this is carbon 1, 2, 3, 4, 1, RO4 dash, this is beta D glucose. Sorry, not glucose. Pyranose or pyranoside. Pwede po. And for this, short abbreviation is beta D gal 1 for beta D GLC. Pwede po beta D gal 1 arrow 4 beta D GLC. Okay. So maltose, by the way, this is incorrect. This should be alpha maltose. No, sorry, that's actually correct because the reducing end is in a beta position. So equatorial, this is axial, this is the anomeric carbons. So you have here this is the non-reducing sugar unit, this is the reducing sugar unit. These are two glucose units, glucose and glucose. So this is gonna be alpha D glucopyranosyl dash 1. This is carbon 1 and this is carbon 4. 1 4 dash beta because it's equatorial at the anomeric carbon. The OH is equatorial at the anomeric carbon. Beta D glucopyranose or pyranoside. Pwede po. Okay. And this is the Haworth projection of this one. So this could also be 1 arrow 4. Okay? So inana siya kadali pag name sa disaccharide. And this should be a practice for you. Okay. How did they come up with this name? Let's just discuss this. It's beta here 1 to 4 because the OH relative to the anomeric carbon, it's equatorial, that's why it's beta, and this is a galactose non-reducing sugar unit. And this is 3,6 anhydro because you dehydrate. Naa siya dehydration step na hitabo between carbon 6 and 3. Like suppose this is okay. So this is okay, so OH let's just say this is the S part. S. This is a sulfite. Okay. So upon dehydration minus water, remove nimo ang water between H and OH here. Loss of water, this becomes an anhydro sugar. Yeah, yeah, this is an anhydro. One is siyang structure, this one. So this is anhydro because it's anhydro relative to carbon 6 and 3. Sorry, not this carbon. Sorry, sorry, sorry. Not this one. Should be here. So na. So that's the structure. Okay, this is the anhydro carbon. This is carbon 6, 5, 4. This is 3, 2, 1. Okay. So this is 3,6 anhydro L galactose 2S because the sulfite group is at carbon 2 and alpha 1 representing this. This should be alpha. Okay. So how does disaccharides form? Disaccharides form by a process called glycosylation. Ang glycosylation, you are just joining or coupling together two monosaccharide units. And in a chemistry, in the basic chemistry, like some biochemistry textbooks, they would show you that this is just a loss of water, right? OH, or let's just say this loss of water minus water, and this now becomes this structure because you are joining carbon 1 with carbon 4. Okay, this is a 1 4 1 4 linkage, glycosidic bond, 1 4 glycosidic bond. Okay, but this happens only in the biological world because we have enzymes, right? Nga kailangan nga very accurate kaayo nga mag-synthesize aning disaccharides. Even daghan kayo siya nucleophilic sites, di ba? It will interrupt with the reaction, but the enzymes will prevent it. Na siya specific kind of orientation nga i-allow lang niya ang pag-react aning duha, ang dehydration step aning duha. So inana ka amazing ang nature. Nature is the best chemist. No one can really defeat nature. Imagine wala siya yung mga synthetic chemicals na ginagamit, but just by using enzymes, it could accurately synthesize this disaccharide easily. But in the lab, kailangan kag protecting group. Na. So ipa-react mo siya. P stands for protecting group. So this is in the lab ha, synthesis in the lab. So okay, ipa-react ra nimo ang kaning anomeric OH. Okay? So in order to do this, kailangan kag activator. A is activator. Ang activator, it could be any oxidant nga mo bind dere. It activates this oxygen. Okay? In such a way nga ang kaning oxygen, ang mechanism would be this, the oxygen out in the presence of an activator here, and the intermediate will be this one. Sorry. This is what you call as an oxocarbenium ion or intermediate. And then this is sugar 1, this is sugar 2, so this is 1, sugar 1 na. So another sugar which is sugar number 2 nga naapoy protecting group. Suppose that this is OP, then OP, OP, then alpha, OP. Okay? Ang isa lang ka OH ang wala na protect. So at carbon 4. So it will attack here and this will bring the octet back to oxygen. So natin kayo ma-form ngayon aning nga bond. This is a glycosidic bond. This is 1,4 glycosidic linkage. So inana ang mechanism of carbohydrate synthesis in the lab. Kailangan ka activator. And also dapat ang OH groups is kailangan mo siya i-protect with various reagents because OH groups could interrupt with the reaction. Nga instead of 1,4 glycosidic bond imong kakakuha could be nga ang connection is between this oxygen and this, or it could be this oxygen hydroxyl or and this, or it could be this primary hydroxyl group with this. So dili ka makakuha sa imong one desired product nga 1,4 glycosidic bond. So that's how you do glycosylation in the lab. Kailangan kag protecting group chemistry. So inani ang mechanism actually sa formation sa disaccharide, and this intermediate also happens in nature itself. So that's how the mechanism works. Like i-eject sa nimo ni una, and then the electrons here will go here and it will furnish an oxycarbenium intermediate, tapos kaning oxygen mo attack dere and bring back the octet to oxygen, and you end up getting this disaccharide. Okay. And also polysaccharides. Polysaccharides are complex sugars made up of a lot of monomeric units. And in glycomics, which is the study of sugars, no? Importante kayo ang symbols. Like there are types of monomers or monosaccharides nga ginatagaan na siya symbol nga convention niya. Like for example, galactose is a yellow circle, blue circle for glucose, green circle for mannose, etc. So that kung i-depict nimo siya ang very complex nga polysaccharide kay nani na yang itsura. Mura na siya kanang mga colorful kay nga mga symbols para easy identification. Okay. So this is the carbohydrate symbols used in glycomics. So very complex. Usually mga biochemists and biologists prefer nila ning inani nga symbols. So here, draw a structure of 75. This is molecule 75. So here, the yellow one pertains to galactose. The square, the blue, is an acetylglucosamine here. That was galactose unit again, and glucose, dark blue circle. And ang linkage nila is beta relative to this, tapos nakakonek siya sa 4 ani nga N-acetylglucosamine. Same thing as this, beta to here and carbon 3 of this galactose. And same thing here, nani na siya. And of course, beta relationship with the protein. Protein maning sp2, a serine residue. Serine. So, step 2 is, kailangan mo siya i-join together. Ang mechanism is, pareho ra gyapon atong akong gidiscuss ani via oxocarbenium intermediate. So that is why it's beta 4, because relative to the anomeric, I will first label the anomeric carbons. So, the O here, the OR group, OR is equatorial. Ba? And this is equatorial, this is equatorial. So that's why it's beta, beta, beta. And also beta here, kay equatorial kaya pala siya. 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1. Okay, 1. So that's why it's beta 4, because beta relative to this non-reducing sugar unit, 1 to 4 linkage. Ang 4 pertains to the carbon 4 of another sugar molecule, okay, which is the reducing. Relative to these two molecules, this is the NRSU or non-reducing sugar unit, and this one is the reducing sugar unit. Okay. Relative to these two molecules, this is the non-reducing sugar unit, and this is the reducing sugar unit. And beta 1,4 linkage gyapon siya. That's why it's beta 1,3. 1,3. That's why it's beta 3 here. And relative to these two, this is the non-reducing sugar unit. This is the reducing sugar unit. So its linkage is 1 to 4. That's why it's beta 4. Relative to the galactose here and glucose here, which is the blue circle, blue circle. Okay, and it's beta to relative to the protein here because equatorial ang bond orientation along the anomeric carbon. And kung i-name mo siya, it's gonna be beta D galactose, 1,4 linkage to beta D. Of course, all of these are beta, so beta ang positioning, ang configuration rather, ng anomeric carbon here. And all the configurations are in dextrorotatory, and these are the specific sugar residues involved in this bonding. So, inanara ang pang-name sa polysaccharides. Pretty simple.